Today we are going to be going over the fourth lesson in the RStudio workshop. This is going to be the longest lesson, but this lesson wraps together everything you've learned in lessons 1 through 3 and puts it into a biological context. So today I have provided you with this document. This is the lesson, the 16-page document. So if you are more of a reading type of learner, everything you need to do this project is written down and everything is explained thoroughly in this. So you really don't have to watch this video unless you learn better by watching someone do it. So I'm going to go over how to do the 3 examples today, and then you will complete the 3 problems, which the 3 problems are similar to the 3 examples. So let's get started. The first thing you want to do is you want to fix your settings. So all you'll do is you'll change the author, the date, and yeah, you can follow these directions here. So let's just jump into the coding. But first, let's read this document. So the goals of this lesson today is you have 3 goals. The first is to learn what the provided data sheets mean in a computational and biological context and be able to effectively filter mass transcriptomic data to identify significant genes. The second lesson goal is to learn what an SIR model is, how the algorithm coerces the population dynamics to evolve over the course of time, and how SIR models can be used in real life. The third lesson goal is to learn how to build a computational model that finds the optimal harvest rate for a fish species dependent on biological parameters specific to the species and understanding the relevance as to why this modeling is important. So let's jump in. So the example involving filtering transcriptomic data uses the disease Systemic Lupus Erythematosus as an example. So SLE is a complex autoimmune disease that can affect various organs and systems in the body, and the exact cause of SLE is not fully understood, but it involves the immune system mistakenly attacking healthy tissues, leading to inflammation and damage. These are common physiological symptoms. The prevalence is pretty low considering the global population, but there's higher incidence in women, particularly during their childbearing years. Treatment for SLE aims to control symptoms, and medications are prescribed to control those symptoms, but lifestyle modifications and regular medical monitoring also play a crucial role in managing the condition. So as research progresses, targeted therapies and personalized treatment approaches hold a high promise for improving the management of SLE and enhancing their quality of life. But the exact etiology of SLE is unknown, and while it's proposed that there's a genetic component, there's no evidence to support this. So today we're going to shed light on this enigmatic condition, and our assignment focuses on investigating potential hereditary factors by analyzing mRNA-seq data. So our goal is to identify genes that exhibit significant upregulation or downregulation in SLE patients compared to healthy individuals. So through this comprehensive analysis, we hope to uncover crucial molecular pathways and genetic signatures associated with SLE, providing valuable insights into the disease's underlying mechanisms and potential therapeutic targets. By advancing our understanding of the genetic basis of SLE, this research endeavor aims to pave the way for more personalized and effective treatment strategies, ultimately improving the lives of individuals affected by this complex autoimmune disorder. So the provided data sheets are crucial for our analysis, and they offer valuable insights into gene expression patterns. these data sheets have already been pre-analyzed from raw data, and it includes 4 essential columns. Well, the first is the gene name. So this is the column that identifies the specific genes that were analyzed in the study. The log2 fold change. The log2 value represents the full change in gene expression between SLE patients and healthy individuals. A positive log2 value indicates higher gene expression in SLE patients compared to healthy individuals, while a negative log2 value indicates lower gene expression in SLE patients. The p-value is the statistical measure that assesses the significance of the differences in gene expression between SLE patients and healthy individuals. A p-value below 0.05 is considered statistically significant. And then the study name. So this column provides a unique identifier for each study from which the data was obtained. And it allows us to differentiate between data sets and maintain clarity during the analysis. So the first data sheet that is online is an example problem sheet. And this contains one study. And for the problem, you'll be looking at the composite data sheet, which contains the data from all the studies that we looked at for this problem. So let's get started and let's take a look at that data sheet, that CSV file, to get a better understanding. So the first thing we want to do is, I'm going to bring over this window, so don't mind me, this is my key for me to type in stuff as I teach you guys, and I pulled this window out so that way I could type on that, but normally your RStudio window is going to look like this, and it's going to have this environment section, and what you're going to do is you're going to say import dataset from base. And then you're going to choose the file that you want. So we want part 1, our lesson 4, part 1, data example CSV. So open. We want to say yes to heading because the CSV file does have headers. And then we're not going to do anything else to it. We are going to change this to just example data. And then we're going to say import. So you're going to take this code that it wrote, copy it, go to your RMD file, paste it in. It's going to be different for everyone because it's dependent on where you stored the CSV file on your computer. But anyways, so this is the example data. So all this data came from PNUSIS 2019. That's the study. But as you can see here, these are all the different genes, log2 fold values, p-values, and we can sort it by the different columns if we need. And yeah. Oh, I guess this is not just how many studies are in this. Is it everyone? I don't remember. Sorry, guys. I can just look. Okay. What about 2019? Oh. What about Arazi? That was another one. No. Okay, so I think I used 2 studies for the example. So that way you're looking at more than one than just one, because the problem that you'll be doing, I think, is 5 studies. Okay, so you imported your data set. The next step is to get rid of the NAs in your data set and coerce them into a data frame. So we're going to name this data frame example, and we're going to use the function naomit. I'm actually going to call this something different. I'm going to call this example data, like this, just because that's what I did before in my answer key. So I'm going to make sure it's the same. Okay, so now we get into the data analysis. So the first thing we want to do is we want to filter the significant log values and p-values into a new data frame with rbind. So what we're going to do is we're going to call this sig filt p. So example. So we're filtering the p-value first. So we're going to use the example data frame. And that's example corrected p value. This is just what so this corrected p value that I'm doing is from this. This is what it's called, corrected p value. And corrected p-value, what we want is we want it to be less than 0.05. And then we're going to put a comma because that indicates that we're grabbing all the rows from the respective column. So know that in syntax, when you're filtering, it's row, comma, column. And we're going to leave nothing in the column thing because we are calling the column and we want all the associated rows. We don't want the other columns. So we're going to do the same thing now, filtering. So now we're going to take the significantly filtered p-values. We're going to take that data set and we're going to filter it, which is significantly upregulated and what's significantly downregulated. So we're going to call this sig filt down dot example. And now instead of grabbing from the example data frame, we're grabbing from the sig filt p example data frame. So sig filt p example, and we want the log to fold change column, and we want it to be less than or equal to -2. And then again, put that that comma. I'm actually going to do something real quick with my windows. I'm going to change something up. So I'm going to put this back in here. And this back in here. And then I'm going to take these out. Hello. There we go. So that way my key is the one that's on my other screen you guys can't see. Wahahaha. Okay. So let's set it up all nice and pretty. So this is your global environment right here. And I think it's important for you to see, so that's why I wanted to do that little change. Okay, so we filtered what was significantly downregulated. So now we're going to filter what's significantly upregulated. So sig filt up dot example equals, again, we're pulling from the sig filt p example sig filt p example, we want log to fold change, and this time it's going to be greater or equal to 2, positive 2. And again, carry that comma. Now we're going to put all this into it. We're going to coerce it all into a data frame. So sig filt example equals rbind and we're just going to do down and up because we filtered the down and up from the P example data set. So we don't have to include R binding the P because the down and up are from the P filtering. Okay. So let's run this and get that data frame. So sig filt example is what we want. So now you'll see all of these values are either -2 or greater. So we only have one that's downregulated, and then the rest are 2 or greater. All right? And there's 37 of them. So the next step is, let's see, we did step 3, so now we're on step 4. So filter insignificant data. So to filter the insignificant data, we want this simply for plotting to, and you'll see this once we do the plotting code, but basically we want the insignificant data to show like how many data points we started with, and then only 37 of them were significant. So we do this by saying insig filt dot example, and we are using the subset function. So we're going to use the example data frame, so our original data frame. We want the log to fold change to be greater or to be less than 2, and we want it to be greater, so we want it to be less than 2 and greater than -2. And at the same time, which is why we're using this up and down thing, we want the corrected P value to be greater than 0.05, because if it's greater than 0.05, it's not significant, right? So that's that. Okay. So the next step is step 5, which is to transform p-values by taking the log 10 ratio for the volcano plot, which is how we're going to visualize this data. So what you have to do is we're going to do this transformation of taking the negative log 10 value for the volcano plot in a for loop. So a for loop, if you remember, is where you have some sort of function you're doing to every row in a column. And so there's so many rows, there's 28,000 to be exact. So that's quite impossible to do one at a time. So instead we do for loops. So we start the for loop syntax by saying for I in one n row. So we're saying we want it to go until the last row of what data set? Well, we want the sig filt, the significant filtered one that we created with rbind. And then we're going to open a curly bracket. So what's in the curly brackets is the function that we're doing. So I want to take all the significant filtered ones, the corrected p-value, and then we say I, and we're referring to each row. So for each row of the column corrected p-value, what I want it to do is I want it to take the negative log 10 of the sig filt example corrected p-value, and again putting I, indicating to do this for every row. So basically what this is saying is it's going to transform this data frame, whatever's in this column of each row, is going to be rewritten over from whatever the corrected p-value is to the log 10, negative log 10 transformed one. And then we're going, so we did this for our significantly filtered ones. So we're going to do another for loop for the insignificant ones. So for I in 1 to n row, now we're doing insignificant. So in sig filt example, open up that curly bracket, call the data set, call the data frame, call the column, and then say I to refer to each row. Then say the transformation. We want the negative log 10 of what? We want it of the insignificant data, specifically the corrected p-value. And we want this for each row. Okay. So if we looked at sig filt example, right, you'll see these are the p-values. This is the order of magnitude they're in. They're in 10 to the negative something, right? And so let's see how this is going to transform our data. So it's been transformed, so we're going to look at it again. And now it's in like regular number, integer, decimal form, right? It's not super crazy small numbers. So this is going to make the data visualization easier, and that's why we did this. So the next thing we want to do is we want to find duplicates, because duplicate genes are significant because it means that they came up many times in the reads, and this is going to be more significant. You're going to get more duplicates when you look at all the data sets together. So we're probably only going to get a couple duplicates because we're only looking at 2 data sets. But when you're looking at the 5 data sets, you'll probably get more duplicates. So what we're going to do is we're going to call it sig duplicates dot example, and we're going to use the table function. So the table function basically just tells you how many of each unique identifier there is. So, and because the gene name is a unique identifier, we're going to use it by that column. So we're going to take the sig filt example, and we want the gene name. And then we want to view this. Sig duplicates example. So let's go ahead and run this code and see what we got. So I'm going to change the frequency. There we go. We have 1 that's a duplicate between the 2 data sets. Yay! The rest, no duplicates. So the last step is to create the volcano plot. So in a volcano plot, it's basically a scatter plot where the log2 fold change values are the x-axis and the negative log 10 transformed corrected p-values are the y-axis. And then we're also going to create vertical lines at the plus and minus 2 on the x-axis, because those indicate the boundaries for significant fold changes. We're also going to color our plots so that way we can see the difference between the insignificant and the significant data points. So let's just start. We start by calling the plotting code, the plotting function. We are first going to do the insignificant data because that is larger than the significant one. So basically the window and parameters of the window that the graph is going to generate depends on the first data set you plot, and you always want to plot your biggest data set first. So that's the x value. We do comma y value. So in sig.filt example, and that's the corrected p-value. We need to do some housekeeping things on the graph, such as giving it a y-axis label, so negative log 10, p-value. We need to give it an x label. That's log 2 fold change, where the ratio is SLE over healthy. Then we need to give it like a main title. So our title is going to be example transcriptomic analysis to identify significantly modulated genes. Now we want to define what color we want these points to show up as by call, and where it's going to be gray, and PCH equals 16, and this is just the size of the dot. And so I chose PCH 16 because there's going to be a lot of dots, so it's kind of small. So now let's plot the significant data, but we want to add it to the same graph. So we do that by, instead of using the plot function, which will create a new window, we use the points function, which will add more points to the same plotting window. So sig.filt example, we want log 2 fold change for the x-axis, and then we want the sig.filt example again, and this time for the y-axis, it's the corrected p-value. And we already defined our ylab, xlab, main, so we don't have to do that again. So we just have to define the color, and we're going to make it red. And we define the size of the points. The last thing we want to do is we want to give it a legend. So we're going to use the legend function, and we want our legend to be on the top left, just because that's going to be where it's easiest viewed. Once you graph your data, you can change where the legend is based off of where it's not hiding any data points. There are going to be two things in our legend. They're either going to be significantly modulated, or they're going to be insignificantly modulated. Did I spell this wrong? Insignificantly. I feel like I didn't. Oh, there we go. There we go. I missed that. See, coding is very finicky on their commas and all that good stuff. So now we need to define the color of the legend. So we're going to use the function fill. And so for the significantly modulated ones, it's red. And then it's gray for the other one. The last thing we're doing is we're adding those vertical lines to define the boundaries of significance. So we use the function abline. V means vertical, and we want it at +2. And we want the color to be black. We need to do this again for the other line. So AB line, the vertical line is at -2, and the color is black. Okay, that's our graph. Let's take a look-see. And there we go. We can pull this out, make it look nicer. It really just doesn't want to look nice right now. But yeah, this is the correct one. I don't know why the fill didn't show up. Oh, I don't have a thing here. And then I need another one. Right? No, I have too many. I only need 2. There we go. So let's run it again. Again, shows how important, there we go, how important your characters are for it. Okay, and that's example 1. Moving on.